Now we are going to learn about immunology. So, living organisms provide ideal habitats in which other organisms can grow. It is not surprising, therefore, that animals are subject to infection by virus, bacteria, protists, fungi, and animal parasites. So, vertebrates have evolved several mechanisms that allow them to recognize and destroy these infectious agents. As a result, vertebrates are able to develop an immunity against invading pathogens. Immunity results from the combined activities of many different cells, some of which patrol the body, whereas others are concentrated in lymphoid organs such as the bone marrow, thymus, spleen, and lymph nodes. Together, these dispersed cells and discrete organs form the body's immune system. The cells of the immune system engage in a type of molecular screening by which they recognize foreign macromolecules, that is, ones whose structure is different from those of the body is known as macromolecules. If foreign material is encountered, the immune system mounts a specific and concerted attack against it. The weapons of the immune system include cells that kill or ingest infected or altered cells, soluble proteins that can neutralize, immobilize, agglutinate, or kill pathogens. Pathogens, in turn, are continually evolving countermechanisms. So pathogens in turn are continually evolving countermechanisms to avoid immune destruction. The fact that humans suffer from a number of chronic infectious diseases such as AIDS caused by a virus, tuberculosis caused by a bacterium, and malaria caused by a protozoan illustrate how our immune system are not always successful in combating these microscopic pathogens. The immune system is also implicated in the body's fight against cancer, but the degree to which the system can recognize and kill cancer cells remains controversial. In some cases, the immune system may mount an inappropriate response that attack the body's own tissues. So it is impossible to cover the entire subject of immunology or immunity in a single chapter. Instead, we will focus on a number of selected aspects that illustrate principles of cell and molecular biology that were discussed. So, however, it's necessary to examine the basic events in body's response to the presence of an intruding microbe. So, we are gonna start learning from basic to advanced level. Okay. So, first, let's have an overview of the immune response. The outer surface of the body and the linings of its internal tracts provide an excellent barrier to prevent penetration by viruses, bacteria, and parasites. If these surface barriers are breached, a series of immune responses are initiated that attempt to contain and then kill the invaders. Immune responses can be divided into two general categories: innate responses, first, innate responses, and adaptive. Adaptive in the sense acquired responses. Adaptive and acquired responses. So, both the types of response depend on the ability of the body to distinguish between materials that are supposed to be there, self, and those that are not there, non self type. So, we can also distinguish two categories of pathogens: those that occur primarily inside a host cell, at viruses, some bacteria, and certain protozoan parasites, and those that occur primarily in the extracellular components of the host, most bacteria and other cellular pathogens. So, different types of immune mechanisms have evolved to combat these two types of infections. Innate response is there, and adaptive or acquired response is also there. So, first we are going to learn about innate response or innate immune responses. Innate immune responses are those the body mounts immediately without requiring previous contact with the microbe. Thus, they provide the body with the first line of defense. An invading pathogen typically makes its initial contact with the innate immune system when it is greeted by a phagocytic cell such as a macrophage or a dendritic cell. So, first line of defense, I guess it's no, first not first line of defense, just first pathogen. That pathogen is greeted by a phagocytic cell such as a macrophage or dendritic cell, whose function is to recognize foreign objects and sound an appropriate alarm. In 1989, Charles Janeway, Jr. of Yale University, published a far-sighted proposal to address the role of the innate immune system both in providing the body with immediate protection from pathogens and in stimulating the cells of the adaptive immune system to become consequently involved in overcoming the threat. We will restrict the present discussion to the innate immune system. So, Janeway proposed that cells of the immune system possess a variety of microbial sensors. Remember this term, microbial sensors. Okay. Microbial sensors that directly recognize certain highly conserved macromolecules that play essential roles in the propagation of viruses or bacteria but are not produced by cells of the body. So, Janeway called such sensors pattern recognition receptors. Pattern recognition receptors. Don't forget this term. Pattern recognition receptors or PRRs. Over the past 2 decades, a number of families of PRRs have been identified, and the most important of which are the toll-like receptors or TLRs. Toll-like receptors. Okay, so which is the only group that will be discussed. So, the discovery of toll-like receptors, which are actually some sort of pattern recognition receptors, is a kind of receptor that the discovery of TLRs came about by way of an interesting and unexpected series of events. The fruit fly Drosophila melanogaster is well known for its iconic status in the field of genetics and also for key contributions to the study of development and neurobiology. But as an invertebrate, Drosophila would not have been considered a likely organism for a major discovery concerning the working of the human immune system. However, in 1996, Julius Hoffmann and colleagues in France identified a mutant fruit fly that was highly susceptible to fungal infections, as is evident from the photograph. The fly pictured is lacking a protein called toll, which had been previously identified as a protein required for the normal development of dorsoventral, that means top bottom polarity of the fly embryo. In fact, toll is the German word for weird, which describes the jumbled appearance of fly embryos lacking a functional toll gene. In addition, they found that toll acted in flies through a pathway involving the transcription factor NFKB, NFKB, which was known to be a key player in the activation of an immune response in vertebrates. It appeared that toll serves as dual function in flies as both director of embryonic polarity and a factor that promote innate immunity to infection. The discovery of toll gene as an important contributor to the fruit fly's defense system led Janeway and colleague Ruslan Medzitov to clone and characterize a human homologue of the toll protein. In addition, they found that the human toll homologue also acted by way of the NFK beta pathway, inducing the expression of a number of immunological effector proteins called cytokines. It was not known, however, what specific role this human toll played in the immune response. Meanwhile, Bruce Brutler and his colleagues at the University of Texas Southwestern Medical Center were searching for mammalian genes that were involved in recognizing bacterial components, particularly a component of the outer membrane of gram-negative bacteria called lipopolysaccharide or LPS. Lipopolysaccharide. They found that a mouse stain that was unable to respond to LPS was lacking a particular gene that encoded an LPS receptor. So, strikingly, this LPS receptor was the same toll homologue TLR4 that had been characterized by Janeway and Medzitov, thus defining its role as a specific bacterial sensor. As it happens, humans express at least 10 functional TLRs, all of which are transmembrane proteins that are found in a variety of different types of cells. So TLRs may be present either on the cell surface where they interact with extracellular microbes or within endosomal lysosomal membrane where they interact with endocytosed microbes. So within the human TLRs family are receptors that recognize the lipopolysaccharide or peptidoglycan components of the bacterial cell wall. The protein flagellin found in bacterial flagella, double-stranded RNA characteristics of replicating viruses, and unmethylated CGP dinucleotides, which are characteristic of bacterial DNA. Activation of TLR by one of these pathogen-derived molecules initiated a signal cascade within the cell that can lead to a variety of protective immune response, including the activation of cells of the adaptive immune system. For this reason, a number of pharmaceutical companies are working on drugs that stimulate TLRs with the aim of enhancing the body's response against stubborn infections such as those caused by hepatitis C virus. Altara, which was approved in 1997 and is prescribed for a number of skin conditions including genital warts, was later discovered to act by stimulating a toll-like receptor. An innate response to invading pathogens are typically accompanied by a process of inflammation at the site of infection, where certain cells and plasma proteins leave the blood vessels and enter the affected tissue. These events are accompanied by local redness, swelling, and fever. Inflammation provides a means for concentrating the body's defensive agents at the site where they are needed. During inflammation, phagocytic cells migrate toward a site of infection in response to chemicals, chemoattractants released at the site. Once these cells recognize, engulf, and destroy the pathogen, inflammation is a dual-edged sword. Although it protects the body against an invading pathogen, if inflammation is not terminated in a timely manner, it can lead to damage to the body's normal tissues and chronic disease. So regulation of inflammation is a complex and poorly understood process involving a balance between pro and anti-inflammatory activities. A number of other mechanisms are also geared to attacking extracellular pathogens. Both epithelial cells and lymphocytes secrete a variety of antimicrobial peptides called deficins. These are some antimicrobial peptides, deficins, which are able to bind to virus, bacteria, or fungi. So that is antimicrobial peptides, which are called deficins. Deficins. Just write that down somewhere. Definsins. I'm sorry for mispronouncing this. Definsins. Definsins. Definsins, which are able to bind to viruses, bacteria, or fungi and bring about their demise. Blood also contains a group of soluble proteins called complement that binds to pathogens, triggering their destruction. In one of the complement pathways, an activated assembly of these proteins perforates the plasma membrane of a bacterial cell, leading to cell lysis and death. So, innate responses against intracellular pathogens such as viruses are targeted primarily against cells that are already infected. So, innate response against intracellular pathogens such as viruses are targeted primarily against cells that are already infected. So, cells infected with certain viruses are recognized by a type of non-specific lymphocyte called natural killer cells. Innate response against intracellular pathogens such as viruses are targeted primarily against cells that are already infected. Once again, I'm going to read this. Innate responses against intracellular pathogens such as viruses are targeted primarily against cells that are already infected. Yeah, that's right. So cells infected with certain viruses are recognized by a type of non-specific lymphocyte called a natural killer cell or NK cell, As its name implies, NK cells act by causing the death of the infected cell. The NK cell induces the infected cell to undergo apoptosis. NK cells can also kill certain types of cancer cells in vitro, and many provide a mechanism for destroying such cells in vivo before they develop into a tumor. So we were discussing about natural killer cells. As the name implies, NK cells act by causing the death of the infected cell. The NK cell induces the infected cell into undergo apoptosis. NK cells can also kill certain types of cancer cells in vitro, and many provide a mechanism for destroying such cells in vivo before they develop into a tumor. Normal, non-infected, and non-malignant cells possesses surface molecules that protect them from attack by NK cells. Another type of innate antiviral response is initiated within the infected cell itself. Virus infected cells produce proteins called type 1 interferons. Interferons is something which are only secreted by virus infected cells. Just remember virus as interferons. This is also a type of innate antiviral response which is initiated within the infected cell itself. Virus infected cells produce proteins called type 1 interferons, IFN alpha and IFN beta, which are secreted into the extracellular space where they bind to the surface of non-infected cells, rendering them resistant to subsequent infection. So what is happening? Which are secreted into the extracellular space where they bind to the surface of non-infected cells, rendering them resistant to subsequent infection. So interferons accomplish this by several means, including activation of a signal transduction pathway that results in phosphorylation and subsequent inactivation of the translation factor EIF2. Cells that have undergone this response cannot synthesize viral proteins that are required for virus replication. So IFN beta may also induce the synthesis of cellular microRNAs that target viral RNA genomes. The innate and adaptive immune systems do not function independently but work closely together to destroy a foreign invader. The dependence of adaptive immune response in prior events orchestrated by cells of the innate system was an important tenet of Janeway's original hypothesis. So most importantly, the same phagocytic cells and NK cells that carry out an immediate innate response are also responsive for initiating the much slower, more specific adaptive immune response. As we will see below, it is the non-specific cells of the innate system that are responsible for activating only those specific cells of the adaptive system that are capable of dealing with the particular threat that is at hand. The next major point include adaptive immune responses. Unlike innate responses, adaptive immune responses require a long period or a lag period during which the immune system gears up for an attack against a foreign agent. So unlike innate response, adaptive immune responses are highly specific and can discriminate between two very similar molecules. For example, in the blood of a person who has just recovered from measles contains antibodies that react with the virus that caused measles, but not with the related virus, such as that which caused mumps. So unlike the innate system, the adaptive system also has a memory, which usually means that the person will not suffer again from the same pathogen later in life. Whereas all animals possess some type of innate immunity against microbes and parasites, only vertebrates are known to mount an adaptive response. There are two broad categories of adaptive immunity: humoral immunity, which is carried out by antibodies. Antibodies are globular, blood-borne proteins of the immunoglobulin superfamily, IGSF. Immunoglobulin superfamily, IGSF. So we just learned humoral immunity, which are carried out by antibodies. Antibodies are globular blood-borne proteins of the immunoglobulin superfamily, IgSF. Then cell-mediated immunity, which is carried out by cells. So both types of adaptive immunity are mediated by lymphocytes, which are nucleated leukocytes, white blood cells that circulate between the blood and lymphoid organs. Humoral immunity is mediated by B lymphocytes that, when activated, differentiate into cells that secrete antibodies. Antibodies are directed primarily against foreign materials that are situated outside the body's cells. Such materials include the protein and polysaccharide components of bacterial cell walls, bacterial toxins, and viral coat proteins. In some cases, antibodies can bind to a bacterial toxin or virus particle and already prevent the agent from entering a host cell. In other cases, antibodies function as a molecular tag that bind to an invading pathogen and mark it for its further destruction. So bacterial cells coated with antibody molecules are rapidly ingested by wandering phagocytes or destroyed by complement molecules carried in the blood. So antibodies are not effective against pathogens that are present inside cells, hence the need for a second type of weapon system. Cell-mediated immunity is carried out by T lymphocytes or T cells that, when activated, can specifically recognize and kill an infected or foreign cell. B and T cells arise from the same type of precursor cell, that's a hematopoietic stem cell, which we will be learning later, that is situated in the bone marrow, but they differentiate along different pathways in different lymphoid organs. Lymphocytes differentiate in the fetal liver or adult bone marrow, whereas T lymphocytes differentiate in the thymus gland, an organ located in the chest that reaches its peak size during childhood. Because of these differences, cell-mediated and humoral immunity can be dissociated to a large extent. For instance, humans suffer from a rare disease called congenital agammaglobulinemia, in which humoral antibody is deficient and cell-mediated immunity is normal. Next is about the clonal selection theory as it applies to B cells, which will be dealt with the help of next video. Thank you for listening.